Morning, dear students. Today's session is all about what happens during embryogenesis, or we can say what happens soon after fertilization, that is post-fertilization. Embryogenesis, I already said, embryogenesis is the process of the complete developmental stages starting from zygote, which is a twin condition zygote, developing into embryo, and then embryo develop into seed. So this is today's topic. We know that soon after the fertilization, yesterday we discussed things like endosperm. In that we discussed about how endosperm useful and what is the structure of the endosperm. Especially we have taken example of coconut, kernel and milk, and even I divided the seeds into two categories based upon the presence and absence of endosperm. Okay, here this is the topic which I covered yesterday, so keep that in your head. Now I move into the zygote development. That is the most important one, where a zygote develops into a seed. Today's session is all about how zygote develop into embryo and embryo into seed. Remember, I already said long back, no more meiosis. From now onwards, every cell division, that means every cell division will be mitosis. Mitosis is the cell division in which they keep the genetic number same in the daughter cells also, but they increase the number of cells. That is the reason why a single zygote, which is not visible, develop into a visible structures like a seed. See, I am holding a seed, a groundnut seed. Let's see what happens soon after fertilization. Look at here, almost everything gets degenerated. Antibodies no longer present. Synapses also gets degenerated. This I already discussed yesterday. And now we're going to discuss about what zygote will become into. It start dividing by mitosis, many cell divisions. I think I already showed in the previous session also. I said they undergo geometric and arithmetic division. Since in our textbook it is given very briefly, so I would like to cover few terms like these are nothing but embryos only. Okay, see, the zygote develops into many mitotic divisions producing thousands and thousands of cells. You can see this is one specific structure of embryo. This is an embryo, and this name of the embryo is called, the structure itself says globular, circular or globe-like globular structure. Sometime soon after continuous mitotic divisions in globular embryo even lead to the heart-shaped embryo also. This heart-shaped embryo develop into a mature embryo. This is actually a dicot embryo, okay, because there are two cotyledons. Now I'll move into the next session where I'll discuss about the structure of embryo. This is the most important, and this is a bit confusing too also. Remember, we are discussing about embryo. That means soon after the zygote, embryo is a structure formed which is like a transition between the seed and the zygote. Now I'm going to talk about the complete structure of the embryo. So what I'm going to do is, in our textbook they given two embryo structural representation. One is a dicot, the other embryo is monocot. Let's see how it look like. Almost same. Both embryos look similar. We're going to discuss about the structure, right? So I'm going to tell you exactly based on the structure of both dicot and monocot in my own manner, so that you can keep it in your head in a proper way. See, I'm going to draw a line here, right over here. I'm going to draw a line, dicot and the monocot, a single line representing a structure called embryonal axis. Embryonal axis, like an axis, like remember top and the bottom, like something like a skeleton. Imagine like a skeleton of a human, like we can call an axis where we can build the rest of the, like if you see this skull, like you have to cover it with the head. If you see limbs, like the bones of limbs, you can cover it with the hands, like that. Imagine this is the skeleton of the both embryo, and this is called embryonal axis in both dicot and monocots. Anything that is present at the top of the embryonal axis here is, this region is called epicotyle. Okay, I think you know epi, top. Same way, anything that is at the posterior part of the embryonal axis, both also. So I'll start with the dicot. This region, not the structure, this region is called hypocotyle. Remember that. Epicotyle and hypocotyle are the proximal and distal end of the embryonal axis. This is a complete structure. It's a tissue made up of like many number of cells. You can see towards the epicotyle region, lateral, this structure that this structure, which is called plumule. Okay, this plumule is nothing but, in future, this is the structure which develop into shoot system. I'll tell you in the upcoming teaching, shoot apex. Same way, towards the hypocotyle region, this structure develop into radical. Radical is the one which will develop into root system. Okay, lateral to both plumule, especially lateral to the plumule, you can see there is a specific structure growing. That structure is called, you can see clearly right over here, I'm drawing neatly. Since there is no space, I'm reducing the structure. This two, like a hands you are observing now, these two are called cotyledon. That is the reason why, since there are two cotyledons present, that's why we call the plants, whoever having these cotyledons, are called dicotyledon. So that means even in the seed also these cotyledons appear. So that's why dicotyledonous seed, dicot plant. The reason why they're called dicot plant is because having cotyledons, two cotyledons raising above the epicotyle, lateral to the plumule. I hope you can understand. And the region, remember epicotyle is not a part, it's a region. Between epicotyle and hypocotyle, there is a region called, this whole region is nothing but tissue, completely made of tissue only. This radical, which is below the hypocotyle region, will have covered with some other structure just below the radical, is called root cap. Root cap. Okay. If you observe the plumule of this structure right over here, this plumule will have shoot origin or primordia. That means you cannot directly call shoot. I mean, like plumule have some cells. These cells are called as coleoptile. Coleoptile. Same way, radical holds some cells which will develop into root system. Initially are called coleorhiza. Okay, keep that in your head. Coleorhiza, coleoptile. Nothing but these are the structures which develop into shoot system, and the coleorhiza develop into root system. Okay, we can say they are the protective layers above the plumule. Same way, coleorhiza is a protective layer above the radicle. You can say right over here. They are present right over here. The same thing will be present here even in the monocot also. But the problem is here the embryonal axis, which we discussed right over here, also almost similar. But the problem, there will be a structure. This is nothing but epicotyle, same like monocot. Epicotyle will hold plumule. Plumule have coleoptile. Same way towards the hypocotyle region also you can see presence of this region is called hypocotyle. This structure which is present at the tip is called a radical, followed by a root cap. Same way, root cap. And then the next structure called coleorhiza. Okay. And since it is a monocot, on the lateral side of the epicotyle the same way. Here also I said on the lateral sides of plumule the same way, lateral sides of the plumule there will be only one cotyledon. Okay, this cotyledon, single cotyledon, and in grasses, especially in grasses, grass plant, this cotyledon is called scutellum. Okay, this cotyledon is called scutellum. So these are the structures almost similar. There will be one specific structure which is given in your textbook called epiblast. Epiblast is a reduced cotyledon. You can see there are two cotyledons raising. Now, since it is a monocot plant, epiblast is the region where there will be a reduced cotyledon, like it stays like that only. It will never further develop into a cotyledon. Okay, so these are the structural differences between a dicot and a monocot. Okay. Remember, if somebody asks you anything like coleorhiza and coleoptile, you are supposed to be giving a meaning. This you should be saying it is a protective sheath around plumule for both also. Coleoptile is a protective sheath above the plumule. Coleorhiza is a protective sheath above the radicle. It will be applied here also. And even now this structure, same structure develop into seed here. Suppose if you take dicotyledonous seed, you remember a dicot seed is having same like embryo attached with cotyledon. So if you take this, this is a seed. Okay, we call seed. But for a biology, for a non-biology student, it is a seed. But for a biology student, it is seed consisting of two parts. One cotyledon, and the other one is embryo. Embryo is nothing but like the same, this embryo, embryonal axis. So embryonal axis having its own structures. Like I already took out, I have broken it into pieces. You can see clearly right over here. Whether you are able to see this clearly or not, but Okay, let me show you. You can see right over here. The one what you're observing, this structure, this is nothing but seed coat. Okay, and then if you observe it a little close, you can see this structure. It may not be that much visible, but I can take a picture and I can send it to you. This structure that you're observing is a embryo. This one, this embryo. And these two, 1 and 2, this structure is nothing but cotyledons. Both are cotyledons only. Okay, I'll show you, and I'll take a picture of it. So it'll be very easy, and it'll be very easy to observe and tell the structures also. Now whatever you're observing is a seed coat. This brown color is called seed coat. Sometime on the seed coat there will be a small opening here, right over here. Yeah, let me continue this. If you observe this structure, no, somewhere there will be an opening. Okay, this opening, a small like opening which is present on the top, is called microphile. I think you remember microphile. If you go back and see in an embryo sac, you can see that integuments are leaving a gap. Now this microphile is the one which is still remain on the seed. So this microphile is meant for absorbing moisture or water, so that in our 11th standard we discussed, entry of water into solids is called imbibition. So water enters, swelling of seed takes place. So as soon as seed swells, you can see there is the cotyledon, and they start developing into a plumule and radicle. Okay, so the microphile is meant for absorption of water into the seed for the germination process. Sometime on the tip, on this region only, there will be a scar. That scar is called hilum. You remember hilum also? Hilum is the structure that joins funicle and the ovule. This, once the funicle is separated, this you can still see the hilum being present. It's like a scar present on the tips of the seed. Okay, so this structure, this is a cotyledon, plumule and the radicle. So both will develop into shoot system, their own shoot system. Radicle develop into root system. You may ask me, sir, what is the purpose of cotyledon? Again I'm going to put this thing straight to you. These cotyledons are in future, as soon as seed germinates, these cotyledons develop into primary leaves. They're not actually leaves, but they are primary leaves. These cotyledons are meant for absorbing the light and do the photosynthesis, produce food, so that the real leaves from the plumule start developing. Once the new leaves start developing, then these cotyledon will fall off, and the complete rest of the things will start developing into a complete new plant. So you can say cotyledons are meant for behaving like initial leaves for photosynthesis, so that the rest of the parts develop into complete new leaves and the new plant. But when you come to the monocots also, if you observe the maize seed, the one which we call corn, the same way you can see right over here the corn. we already discussed in our 11th standard also. You can see the male seed is also having, I already said, like they're made up of endosperm, right? Endospermic seed. Endosperm is still attached to the real embryo or the seed. So endosperm, you can see the scutellum. I said no, scutellum. Scutellum is this part which is a monocotyledon, single cotyledon. Same way all these structures. Plumule, coleoptile. What is the difference between this and this? This is a seed and this is an embryo. Okay, so you can see clearly right over here. Okay, so that is what the structure of monocot seed and dicot seed. If we move a little further, there is one specific structure we would really like to know. This is called the seed. Sometimes even some plants like black pepper, beet, still holds the nucellus being attached to the seed itself. This nucellus, which is present all the way from the megasporogenesis till the seed formation, is called perisperm. Okay. Now what is perisperm actually? Now these plants have their own seeds. Like suppose this is the seed of black pepper and a beet. Apart from endosperm, sometimes black pepper and beet do have still a little tissue present around it, which is naturally, it's a nucellus. You know, if you go back and see nucellus, what is nucellus? If you go back into the past, you can see the ovule structure. In that there is embryo sac. Embryo sac is surrounded by nucellus. You remember, no? Nucellus is the tissue that provide nourishment to the megaspore mother cell. The same tissue still remain even after fertilization. Even after fertilization also, it's still present. I think you know, right? Embryo sac is the one which will develop into seed. So that's why the seed of black pepper and beet will be surrounded by persistent, simple answer, persistent nucellus is called perisperm. This makes a little sense to you. Now, if anybody asks you perisperm, remember persistent nucellus present around the seed in black pepper and beet is called perisperm. Okay? Fine. Tomorrow we'll discuss more about fruit. Okay? And few things related to seed also. Thank you and have a nice day.